Welcome to everybody. Good afternoon or good evening for those coming from Europe. So we are truly honored to have been invited to participate in this wonderful webinar. I must say that IVF meeting has really made us full united and connected during these long weeks of lockdown. And now being part of this meeting, fantastic tonight, makes us very proud of this. And so SIERR wants to thank the organizers, and in particular Jacques Cohen and James Palmer for this opportunity. But if we go through what SIERR is, let me spend a couple of words. We are a society of at the moment, of more than 400 members, active members. You see here, this is a picture, a puzzle of all the members that participate the Embryologist Day in 2018. So if we go through the acronym, SIERR is the Italian society, so a non-profit Italian scientific society founded in 1998, with the aim to promote the development of clinical embryology, strictly related to assisted reproduction and basic and applied research. So in the name, there is everything we would like to be and we would like to transmit. So to pursue this goal, our hashtag is "We are growing in SIERR." So we are growing in SIERR, and to grow together, we have to create connections, make new collaborations, caring about our members. And for example, we are the first Italian scientific society that is offering and providing insurance and legal assistance to all the members included in its fee. So this is very important for our members. But to share knowledge, we also are involved in teaching. We are involved in many university master's PhD courses. We write position statements. We developed scientific projects like the one we are publishing right now on the endocrine disruptors. And most of everything, we would like to spread our knowledge between members, but because we like to create connections also with professionals of other disciplines. And we like to make new collaborations like the one we are doing right now with you, and the one with the foreign societies. In our society, there is also a group of SIERR Beyond the Borders, that is a group of Italian scientists that are working in different disciplines in different parts of the world. So to pursue this desire to spread and to grow together in SIERR, we were thrilled and amazed to participate in the organization of this meeting, The Future of Embryology, and the subtitle is Technological Avant-Garde in Embryo Selections, just because we believe that we did a very good job in these 40 years of IVF, but we have to do more in the next future. And the technology will help us and will provide the instrument to perform every day a little better than the previous day. So it's my pleasure to give the floor to my co-chair, Catello Scarica, the vice president of our society, and to introduce these two outstanding speakers, Marcos Meseguer and Maurizio Poli, that are exactly the representation of what we want the society to be international and to share knowledge with other societies like the SIERR, and to share knowledge with different professional figures that can enhance and improve our knowledge, our society, and at the end of the day, the everyday life of clinical embryologists. So thank you, Catello.
Thank you, Lucia. Thank you once again to the organizer of the IVF meeting and to the moderator speakers of this session. And on behalf of SIERR, I would like to thank in particular also Francesca Klinger and Danilo Cimadomo, that are scientific coordinators of SIERR and also members of our executive committee, and that made this webinar possible. So thank you. Now, going through the topic of this webinar, as Lucia told before, we are embryologists. So as Embryology Society, we are facing IVF laboratory problems, everyday problems, based on the present, and trying to improve and standardize what our members do in everyday practice. We try to support them, and for instance, we are performing a study about the morphological embryo assessment to enhance inter and intra-operators concordance, trying to avoid variability among operators. And this is what we do for the present of the IVF. On the other hand, we are strongly convinced that innovations and further knowledge will improve, of course, IVF laboratory performances. And for this reason, we have focused the topic of this webinar on possible next evidence-based applications in the IVF in the light of technological evolution. So we will speak about the future of embryology. It's a great pleasure to introduce to you Dr. Marcos Meseguer. He's the scientific supervisor of EVRMA of Valencia, and he's a great expert of IVF, as we know. And recently, he's interested in artificial intelligence in reproductive medicine. He will share with us his experience, and we are very proud and glad that he accepted our invitation to join us today, also for the feelings of closeness that we have with him, and for the collaboration between our society and the Spanish society as a beer. So Marcos, thanks again, and let's start with your presentation.
Thank you very much for the introduction. Buonanotte, Forza Italia. Grazie per la invitazione. It's un placer, it's my pleasure to be here, and I would like to thank especially Danilo Cimadomo and Antonio Capalbo, which are also my friends in the society, sharing this opportunity with me, and with the Spanish experience also with embryologists here in SIERR, which definitely also are becoming a very good and important society. Well, the members of the Italian Society of Embryology asked me to talk about the embryo grading in the laboratory, and I'm going to move forward in that field. This is the agenda of the topics of today. I would like to introduce the concept of precision embryology, how we are able to identify markers of embryo quality. And also we'd like to talk about the automation of embryo evaluation with 3 softwares that we have been testing in IBI in Valencia: the Connect and Assess, the Guided Annotations Kit Score, and the EVA Extend. Also, we'd like to show the experience that we have comparing time-lapse incubators with conventional incubators, our last experience with this technology in Valencia, and also a little bit about artificial intelligence applied to embryos. So the first topic will be precision embryology. I would like to introduce this idea, which I love it, which is the possibility to identify new markers of embryo selection. And to do that, I have bring a video that has done one of my colleagues, embryologist, Lorena. She's working doing the PhD in Valencia, and she's making this video to make you an example of the amount of features of the embryo, starting from the zygote and going to the evolution of the embryo from pronuclear stage to blastocyst, and how you are being able not only to identify new parameters that are affecting or just the appearance of the pronuclear, the disappearance, also you can perform a lot of quantifications. In particular, they are doing a follow-up of the time in which the pronuclear appear, and also the time in which the pronuclear is fading, and how this movement can be measured just taking an advantage of all the tools that are in this type of software. This is the embryo viewer of the embryoscope, and how you can try to measure all these parameters. So now we just move forward in the embryo development to the cleavage stages and all the timings that are annotated in the software, in this case manually. So it takes a lot of time to do the annotations for only one single embryo. We move to this precision embryology, trying to identify new markers of embryo selection. And when we move from morula to blastocyst, we may start to identify the blastomeres also. And those blastomeres that we are seeing here, we can try to follow, and we can try to let's say, not only identify one blastomere, also to identify the cell cleavage of a blastomere of the trophoctoderm and try to quantify the duration of the cell cycle of a trophoctoderm cell, which is not easy. You need a very high quality of the embryo, and you move also with different focal planes in order to follow this, this blastomere, and how we can be able to detect the next cleavage and try to do a calculation of the duration of the cell cycle of trophoctoderm. If we keep moving forward in the evaluation of the blastocyst, we may end up with the last image of embryo development, and doing that, we may also calculate which is the final diameter of a blastocyst. So we can make a quantification, again, using the drawing tools of this software. We can make a quantification of the final diameter of the blastocyst, and also not only the final diameter of the blastocyst, we can also quantify which is the area of the inner cell mass, for example. So it's a good example of the amount of features of the embryo we can take into consideration when we do this type of evaluation. So put you in context and trying to see an example of the relationship between these parameters that we measure and are possible to be quantified and not subjective, we can see a clear relationship between the expansion of the blastocyst and the implantation potential. So you have a very quantitative data that can be easily linked with the outcome, and also the area of the inner cell mass. You can see how the area of the inner cell mass can be linked also with the outcome, with some limits that can define a better or worse outcome. So it's, let's say, like a good way to quantify the quality of the blastocyst using parameters that we can measure. Another important feature that we can analyze, and you can follow this video, is the behavior of the blastocyst after warming. We can measure which was the initial area that the blastocyst have after warming, that you know that is collapsed, and we need to give to the embryo around 4 hours for the process of re-expansion. And also we can measure the thickness of the zona pellucida at the beginning of the warming, and we can wait 4 hours and do a follow-up of the behavior of this blastocyst after full re-expansion during these 4 hours or not. And then also which was the final area of the blastocyst after 4 hours of evaluation of the blastocyst, and also which was the final thickness of the zona pellucida. So it's another way of evaluation of the embryo after warming that may potentially be useful for knowing the outcome of that embryo. And this is the case, and this is data that we published in fertility and sterility a couple of years ago. We were able, just measuring this initial area of the blastocyst and also the final area of the blastocyst, together with the thickness of the zona pellucida, and we were able to link these parameters that are totally objective with the outcome of these blastocysts after warming. So clearly, is a good example of how we can use this technology to quantify the quality of the blastocyst at different times of the embryo development, and this in this particular case, in the blastocyst warming. Even we have seen that the embryos that normally implant takes less time to start the process of re-expansion than the embryos that do not implant. So even we can quantify how long does it take for the embryo to start the process of re-expansion. So also we can analyze the embryo automatically. So I have bring you here the 3 examples of 3 software that we have been using in IBI Valencia, and we have been testing and evaluating about how we can do the automation of embryo evaluation. One of the software that we have been trying is the Gary Connect and Assess, with the 2 versions, the Assess 1.3 and the Assess 2.0. These 2 versions of the Gary are able to do annotations manually or automatically. If the annotations are done manually, every step of embryo development should be detected by the embryologist and annotated. But also we have the option of using the Assess 2.0 that is doing all these annotations automatically. So each event of embryo development is detected automatically by the embryo without any intervention of the embryologist. So we have these 2 options, and we're just wondering if these timings that are estimated by the software are similar to those estimated by the embryologist. Because we can face 2 situations. We face the situation that, for example, the timing that is annotated by the embryologist and the timing annotated by the Jerry are very similar, or that the timings annotated by the embryologist and the timings annotated by this Jerry software are very different, and then we have a problem. So we presented this data the last year in SIERR, and we're just taking a look to the timings and the difference between the timings annotated by the embryologist and the SS 2.0. We observed that the difference was close to 0, and only in those timings that are subjective, we start to see a larger difference between the annotations performed by ourselves and the machine and the SS 2.0. But for example, if we take a look to all the annotations performed by the embryologist in IBI Valencia for time of 6 cleavage to 6 cells and the annotations performed by SS 2.0, and we merge those, we see that are very, very similar. So once we have all these automatic annotations, also we have a warning from the software that is helping us to, let's say, take a look to those annotations that are out of the normal range that was published by some of the publications already reported, and then take a look to that timing and maybe modify if it's not correctly annotated. So once we do the annotation manually or automatically, we're just wondering, okay, if we perform the annotations automatically or manually, and we use this morphokinetic algorithm that allow us to grade the embryos, how we can see if this embryo grading performed automatically or manually are related with the clinical outcome. We'll select the same embryo for transfer. Well, in order to do that, we take for reference one paper that we published together with Danilo and with Laura Rienzi in Italy with Genera Roma. And in that particular paper, we just pay attention with the time of compaction. And this time of compaction with a limit in 18 hours, we're defining embryos with more or less chance to implant. If you take a look to the annotations performed by the embryologist and this SS 2.0, the proportion of embryos in each category is different if the annotation is performed by the embryologist or by the SS 2.0. So if they were annotated differently, the time is annotated different, then the grading may affect the clinical result prediction. And this is the case. If you take a look to the implantation rates of the embryos that have the compaction before or after 80 hours, there is almost no difference in the implantation potential between these two types of embryos depending on the time of compaction. But there were differences between the annotations performed from the SS 2.0 and the implantation potential in comparison with the embryologist. So it looks that in order to do the annotations and in order to do the algorithm for embryo selection, especially for variables that are subjective, this software is more helpful in this type of process. So that's one example of this standardization of the annotations. We have been working with other software. This is the Kids Core Day 5 that is performing also annotations with a guided software. So all the timings are presumably detected by the software, and we just need to confirm each time that is relevant for the time of cleavages up to 6 cells, also the time of blastulation, the time of starting blastulation, or even the quality of the inner cell mass on top of them that is annotated automatically by the software. There was 2 versions of this software that were grading the embryos from 1 to 9.9. The version 2 and version 3 has some difference in the variables that are used, but in all of them, we are ended up with a grading of our blastocysts, which is quite interesting because, I mean, we don't need to do any grading. The software is doing the grading for us from 1 to 9.9, and we can choose this current score in order to select our blastocysts for transfer. In particular, in many cases in which the blastocysts look very similar by morphology, which is the case of this patient. So we have been trying to correlate this information with the outcome. We have seen a relationship between live birth rate and this KID score. This is the version 2 and version 3. This is the number of embryos that were transferred. And if you take a look, if we split the KID score in 4 categories with similar proportions, we observe a relationship between the live birth rate and this KID score from 20% to 48.6%, where the grading is over 7.5. Even if we take a look to the version 2 and 3 and the euploidy rate, we see that the categories that we created, in particular with the version 3, from 3.4 to 6.5, there is a linear grading and relationship between the proportion of euploid embryos and this KID score. So it's a nice relationship also with the chance to have an euploid embryo. The other algorithm that we have tested is the extend algorithm. This is an algorithm totally automatic that is doing annotations of the embryos from 2 to 3, 3 to 4 cells. It's also performing a post-P3 analysis from 4 cells to day 3, and also is counting the number of cells and is giving also the information about the age of the patient. I have bring you here a video of the system, how it works. We have all the embryos from our patients, and with this EVA grading, all the annotations are performed automatically. These timings that I told you, the time of clearance to 2 and 3 cells, and also is giving us a number from 1 to 5 automatically. So each one of the blastocysts that we have, we can take a look to the morphology, but we end up with a number from 1 to 5 that is performing this automatic classification. And we may potentially use this grading for the selection of our blastocysts that you can see here with those numbers. So how this information provided by this software is related with the outcome. Again, here we have been working in this algorithm. This is using dark field, which is different from what the embryologists are used to do to evaluate the embryos. This is data that is part of the PhD one of our embryologists, and we try to validate this software. First of all, taking a look to the proportion of categories that are performed by the software is more than 5,000 embryos that we have been analyzed. And the proportion is more or less similar for each one of the categories. And there was a nice link between these categories performed on day 3 and the proportion of viable blastocysts. Additionally, also to that, we take a look to the blastulation in day 5 and day 5 plus day 6. Those are the total number of embryos analyzed, more than 5,000 blastocysts. And you see how the proportion of blastulation was linearly related with this automatic categorization from 1 to 5. And regarding implantation potential, also we take a look to close to 800 embryos transfer from our program, mostly with single embryo transfer. And you see also that there was a relationship between these categories and the implantation potential that was linear. Finally, we take a look in our PGT program if there was a link between these categories and the proportion of blastocysts that were available for biopsy. And again, in this particular study that we presented last year in SIERR, we observe a linear relationship between this automatic categorization of the embryos in day 3 and the probabilities to perform a biopsy on day 5 or day 6 to that blastocyst, from 74% to only 17%. So the next thing that I would like to talk is about the comparison between time lapse and conventional incubators. There is a lot of randomized trials, retrospective studies. We have participated in some of them, but I would like to update you in the last couple of years. In Valencia, we have a very particular environment because we work with benchtop incubators. We have Aztec benchtop incubators, but also we have the Jerry, we have the EmbryoScope Plus, and we have the EmbryoScope standard EmbryoScope, and we have the conventional incubation like a reference. It's a very particular environment because we are a big clinic. In the last couple of years, we perform around 9,000 ICSI cycles during 2 consecutive years, more than 26,000 embryos analyzed, and close to 10,000 transferred. And just we take a look to the results in general in our population in these 2 years. And just taking a look to the numbers, we still have better outcome, around 60% of implantation rate when we use time-lapse incubators in comparison with standard incubators. So in our environment, in our clinic, I mean, we have performed, and we are presenting this data this year in SIERR, a little bit more complex, but we still have better results by using this technology in comparison with standard incubators. I would like to introduce the concept of oxidative stress. It's not nothing new, but we have been testing another hardware called TCL or Fertissimo. It's a software that has been developed in Israel. It's basically measuring reactive oxygen species in the embryos. We know that the embryos produce reactive oxygen species. We know that are important for the cellular metabolism, are important for the function, but also we know that these levels may impact also the cell, may produce changes in the membrane, the lipids, in the proteins, and may affect the outcome. So we have been testing this software. It's a very simple way to do it. This software is having this hardware, is having cuvettes in which we put the media in which the embryos has been growing. So we take 50 microliters of the media in which the embryos have been growing in the slides of the EmbryoScope or in the Jerry. We put in these dishes. These dishes are moving from one station to the other. First of all, in the station 1 is performing a shaking to obtain a unified layer. Then it's moving to a second station in which the sample is drying using a vacuum. From this second station is moving to a 3-1, which the sample is heated and scanned. And then we get the emission of photons per second at 80 degrees that are related with the oxidative stress. This is data that we published last year in fertility and sterility. And basically from this hardware, what we get is 3 variables, H2, H1, H2, and H3, that are related with the oxidation of the media or the reactive oxygen species that are in the media, and taken at different timings. We have been using the combination of artificial intelligence and time lapse and the information of oxidation. We have performed a training of artificial neural network. We have performed a model, a simple model of artificial neural network, trying to see if the combination of the information from time lapse and information from oxidative stress may improve the accuracy in the prediction of live birth. And we end up that we were able to create a model that, combining morphokinetics and the oxidation, we are reaching higher predictive abilities for embryo selection than only morphokinetics. My point here, I mean, we have a lot of data in this topic, but my point here is that adding this value of oxidation to the morphokinetics, we may end up with a better prediction of the outcome of that cycle or the outcome of that embryo that we're going to select. Finally, we'd like to talk a little bit about artificial intelligence. We can spend like, I don't know, many hours talk about this topic, and we have been working a lot with many groups, but I would just like to focus a little bit in the concept. The concept of artificial intelligence is relatively simple. We just provide information, blastocyst images, morphokinetics, maybe patient demographics. Then the concept of artificial intelligence is create an artificial neural network, ANN, with several layers in which the information is collected, is split, is shared, and then we end up with an output layer in which we may potentially predict blastocyst morphology, I mean, substitute the embryologists to predict morphology, predict implantation, or predict live birth, or even predict miscarriage. I have bring you here a video on how this potential software may work. This is a prototype performed by University of Sao Paulo. There is a group of mathematicians there that has been working with images of the embryoscope. They have been developing artificial intelligence networks, and then they end up with a prediction. I mean, you have 4 blastocysts, very nice, good looking, and the software is telling you which one of them has more abilities to reach live birth. If we take a look to each one of the images, we can see that the embryo has been analyzed and the software has learned to identify where is the blastocyst, where is the inner cell mass, and where is the trophotodem, and then quantify the quality. This has been performed with images of the embryoscope, but also this has been tested with images of the gerries. So it's the first time, because at the end, this software learns from one type of time lapse, but the idea is to be able to be used in all different type of time lapse machines. So in this case, you see that the image of the blastocyst is different because the picture taken by the gerry is different from the embryoscope. So we need to teach the software to identify the embryo in a different image coming from a different time lapse. And also we need to help or teach the computer to identify or to locate where is the blastocyst in this well that is different from the well of the embryoscope or a well of another time lapse system. So that takes time, but at the end, you end up with a system that again is able to locate the embryo, locate the trophotodem, locate the inner cell mass, and then perform a prediction of the outcome. So we have been working with this software, and still the data is very preliminary, and we have been presented some of the data in several congresses in the last couple of years. But what I can share with you with the studies that we have been performing is actually or using only morphokinetic data or using morphology, and still with limited sample size, and training the software and then perform tests, the accuracy of the prediction of live birth is extremely high. The numbers are still limited, but the technology looks very promising because we are potentially being able to combine the knowledge of artificial intelligence to improve our selection. So finally, I would like to talk a little bit about the possibility of artificial intelligence to discover or quantify new aspects of the embryo development. For example, in this case, this is a software that was developed for a company called IVF in Israel. They just quantify the irregularities of the cell membrane and identify like green those parts of the membrane that are good and red those that are wrinkly, and try to link this aspect of the zygote to the outcome in the first 24 hours. So I have bring you here a video trying to show you how you can identify a lot of parameters of the embryo that are potentially during embryo development, that here are represented by colors, that potentially may be integrated in a prediction of the outcome. In this particular case, they are only working with the first 24 hours, trying to predict what will be the outcome of that embryo. We are going to present part of this data this year in SIERR online. So to conclude, about what we have seen today, first of all, I would like to underline the importance of the observation that the embryologists are performing and how time-lapse are allowing us to precise definition of many relevant events of embryo development for embryo selection. For example, blastocyst diameter. So time-lapse is helping us to learn a lot about embryology. And also, we just can take a look to the expansion of the blastocyst after deritrification, after warming, and how this is quite linked with the outcome. Time-lapse has been able to introduce us the process of automation by first time for embryo evaluation, by fully automatic morphokinetic and morphology annotations, by performing software like Assess 2.0 that apparently are improving the selection or the precision performed by morphokinetics from the embryologist, the KID score algorithm for embryo selection that is also successfully linked with the blastocyst morphology and is linked with the outcome and also with aneuploidy, and the direct correlation that we have seen between the extent classification and the implantation potential. We have seen that time-lapse incubators in comparison with the standard are improving the outcome. We have better results. And also that how oxidative stress may be potentially be a marker of metabolism marker and complementary method for embryo selection. Finally, talking a little bit about artificial intelligence, I believe that this matching embryologists to qualify human blastocysts, the application of this technology to the classification of human blastocysts is relatively reliable and is non-invasive. It's a promising diagnostic tool for prediction of outcome, and also apparently assess very fast and is continually learning. So let's say that this type of software with the time will be improved because it's learning every time with more information. Finally, I would like to show you the team of embryologists that are helping me with this project, in particular Sonia, Carmela, and Belen, that are working with me like senior embryologists in Valencia. Also, all the team of junior embryologists and PhD students that are working in time-lapse research and artificial intelligence. These four nice, good-looking embryologists, Raquel, Lucia, Lorena, Maria Angeles. Also, I have a lot of people helping us with artificial intelligence from Sao Paulo, from University of Sao Paulo, and also from Israel, from IIBF. And also, we'd like to thank everyone for the attention of this meeting.
Thank you so much, Marcos, for your great presentation. And I want to remember to the attendees that the question time is at the end of the two presentations. Please continue to use the ask a question and answer section and not the chat, please. And International IVF Initiative has a talented team who is working behind the scene to post your question to our speaker. So I encourage you only to use question and answer section to conference service. I will introduce you the next speaker, Maurizio Poli. Dr. Maurizio Poli is a member of the scientific committee of SIERR, and actually is a clinical embryologist and reproductive scientist involved as a scientific supervisor at Genomics Italy. He's also a co-founder of Genomics, which is a company developing proteomics-based methodologies for reproductive medicine. He is the first who performed the blastocentesis for proteomic analysis, analyzing the blastocoel fluid, and he has a great experience on non-invasive assessment of embryo developmental competence. So his presentation is about the past, present, and future strategies for enhanced assessment of embryo's genome and reproductive competence in IVF. So Maurizio, thank you, and let's start with your presentation.
Dr. Scarica, thank you for the introduction. Hello Hello, everyone. Thanks for connecting. My name is Maurizio Poli, and I'm connecting from Haarlem, a small town outside Amsterdam in the Netherlands. First of all, I would like to thank the International IVF Initiative for hosting such an informative platform. I enjoyed it very much during these times of home confinement, and I hope they will continue to publish new webinars while things are getting better. Thanks to SIERR as well for the invitation to give this presentation. And this is going to be a short overview of the current and future possibilities that genomics and other omics will offer in the context of embryo selection and deselection. So I very much enjoyed Marco's presentation and also other presentations presented on this platform about machine learning and artificial intelligence. I think there's great potential, and everyone agrees that the time-lapse incubators are a great addition to the IVF lab because they provide a stable and uninterrupted embryo culture condition and generate a great amount of data with no need for biochemical analysis. So costs are only limited to the embryologists spending their time on the computer. But on the other side, this may be a limitation as well, because complementary and biological parameters, I think, that will need to be considered if we want to develop a highly effective prognostic tool. So some parameters, and I show them here, can be used for identification of biomarkers, and these could be genomics, for example, morphokinetics for sure, and all the plethora of omics, including proteins, metabolites, and transcripts. And others instead may be used, other parameters may be used to normalize and characterize subgroups of patients that we are including in our study. And these involve pharma treatment for sure, but also detailed laboratory parameters, such as how the processes that are performed in the lab, culture conditions, and also culture media composition that is sometimes too often neglected. So through the acquisition and digital recording of high quality data from multiple sources, this will become more crucial to generate a comprehensive picture of the human embryo's biological characteristics, but also differences between them, metabolic preference, and reproductive competence ultimately, and allow the development of data mining and machine learning platforms that could enable true personalized medicine that can make an impact in a meaningful way. So when we talk about embryo assessment, the first consideration that we need to make is whether we want to assess constitution. And in this case, we're talking about the long-term impact on health of the ensuing child. And it is information that is usually invariable over time and affects the core of the flow of the biological information. What I'm talking about is DNA. By assessing DNA, we can understand whether our embryo has a correct number of chromosomes or if it has the presence of pathogenic variants, and possibly define the multifactorial disease risk that it carries. On the other hand, if we want to define the function and we want to predict the ability of an embryo to generate specific short-term outcomes, such as the ability to develop a blastocyst to the blastocyst stage or even implant to the endometrium, we need to investigate molecular characteristics that are closer to the phenotype, like transcripts, proteins, and metabolite changes, and provide a real-time picture of embryo status while it is affected by the specific environment that it grows within. So for this reason, I believe that omics and morphokinetics may provide a better approach to determine physiological status. I share this view with many other people, and some of you for sure, that the future of IVF runs through genetics. And this is particularly true if we think how PGTM, so pre-implantation genetic testing for monogenic disease, has been so effective in preventing the transmission of inheritable single gene disorders. And PGTM was the first application of pre-implantation genetics, and surely it has, it's the only undebated application of PGT. There is a strong consensus behind it, and because of its clear benefits, and it is so strong that also countries with an historical aversion towards chromosomal screening still allow PGT-M in their labs. And I'm talking about Germany or the Netherlands, for example. On the other hand, PGT-A, so pre-implantation genetic testing for aneuploidies, is still widely debated. Its main application is in those cases where there's an increased risk of producing embryos with an abnormal set of chromosomes. And it's conventional to think about advanced maternal age patients and patients that have experienced recurrent pregnancy loss. So the aim of PGT-A is to avoid the transfer of embryos that we already know do not implant, or if they do, they will inevitably generate a negative outcome. And this has been shown by several studies worldwide that PGT-A can help in the reduction of pregnancy loss and the minimization of the risk of the birth of a chromosomally abnormal child, still maintaining the same delivery rate per cycle. So I know many may be interested in mosaicism, but I'm not going to address the elephant in the PGT-A room tonight, and that's because I'm aware that more webinars will be available shortly in the next few days. So please link to them if you're interested in mosaicism. So discussing new developments in PGT-A, one of the interesting ones is the combination of PGT-M and PGT-A in a single assessment. So this is directed towards PGT-M patients that want to ensure not only that the embryo that is transferred is not affected by the inheritable genetic condition of the parent, but they also want to test for aneuploidy. So this approach to me makes much sense because, first of all, the embryo biopsy has already been performed, and the embryo most probably is going to be cryopreserved as standard in PGT-M cases. Therefore, it makes sense to make sure that not only there's not the mutation, pathogenic mutation, but also that the embryo can implant and go on to form viable pregnancy. On the other hand, expanded PGT, also called the ePGT, is more tailored to PGT-A patients that using a similar technology, we can assess not only the chromosomal status, but also additional genetic features. And these include the de novo mutations in the embryo or in the that happen in the embryo or in the germinal line of the parent. This is quite interesting if we think that it's been demonstrated that a significant proportion of neurological conditions in children derive from dominant or X-linked de novo mutations. So it could be very interesting and meaningful add-on to our PGT system. Also, through ePGT, it is possible to assess polygenic traits such as diabetes type 1 or specific susceptibility to certain cancers, and as well as genes involved in embryo developmental insufficiency. And I'm gonna touch about that a bit later when I talk about genome sequencing. So another trophectoderm biopsy-based technological development for evaluation of embryos competence involves the simultaneous PGT-A and transcriptome assessment from a single biopsy specimen. This application is not yet in the clinical context, it's not yet applied, but the technical quality control parameters appear to be fairly robust with 100% concordance between PGT-AT and the standard PGT-A methods for what concerns a chromosomal status. And this is according to this recent publication. However, there's no correlation between the transcriptome features and the clinical outcome yet. However, it will be interesting to see how this technology will develop in the future and how the results will come in when they're going to apply to a clinical context. Source of debate regarding PGTA, probably the main one, is the potential mechanical damage enforced by the biopsy procedure on the embryo. And although several studies have showed that the biopsy has very limited impact if it is performed by experienced operators, there is always the risk and the chance of the occasional difficult case, or for labs that are implementing PGT for the first time, they have to face a quite steep learning curve on doing the biopsies, and therefore there will be a period of suboptimal results. And that's why several groups have invested resources in developing non-invasive approaches for PGTA. And my colleagues from Genomics Spain have just published the largest data set about this topic. And it appears that the non-invasive PGTA can provide additional data to morphology in terms of which embryo to prioritize for transfer. However, sensitivity and positive predictive values are still inferior compared to biopsy-based methods. But however, it looks like there are differences between laboratories, and this suggests that the technical improvements could be made in the future. And if they are achieved, this technology has surely the potential to be employed routinely in the IVF lab. So continuing on non-invasive approaches, I'm going to move to the other omics. So metabolomics and proteomics have been employed since the 90s in IVF. And although very promising because of the molecules that they target, they have only been used in research setting with no real clinical application for IVF just yet. They have the potential to assess embryo metabolic activity and investigate its acute physiological state, also taking into consideration the embryo response to the environment that it develops in. And this is, to me, is quite important. It's not the selection process, but rather it would provide a grading system for prioritization of the embryo to transfer. These type of omics, fairly based on biochemistry, they face quite specific challenges compared to genetics, for example. Differently from DNA, proteins cannot be amplified, and the detection has to happen in their native concentration, which can span several orders of magnitude. Also, absolute protein production from the embryo is really, really small, and it requires extremely sensitive instruments to perform it. Surely in the recent years, development of instruments such as mass spectrometry have made possible to detect such small amounts of compounds. However, there are also confounding factors and contaminants, especially in the culture media, that can be detrimental to these kind of assessments. For example, albumin is present in a very high abundance and can mask those proteins that are embryo-derived and are secreted in very, very tiny concentrations. Also, in culture media, there are contaminants that are uncontrolled and undetected during QC processes. And therefore, this produces a batch inconsistency in the compounds that are present within the media. And surely, or it is possible that these differences can have effects on embryo developmental features that are not taken into consideration when we use other types of investigation methods. So I believe that detailed quality control of the media will be required in the future if we want to standardize the formulation and provide a consistent environment for embryo assessment and allow cross-evaluation, even for morphokinetics. Despite all these limitations in proteomics and metabolomics, my colleagues and I showed a few years back that specific protein biomarkers can be consistently identified in human blastocysts, which it's arguably the smallest specimen that one can investigate with a mass spectrometer. And we demonstrated that this technology is now available and at a sufficient level for clinical investigation, also for IVF. To conclude, I would like to go back to genetics, this time to preconception genomics, because I believe that this is a methodology that will take a lot of space in the future of IVF. And many of you may be already familiar with the carrier screening because it's been used since the 70s in various technological forms, and not always with genetics, also with biochemistry in the past. So carrier screening assesses the presence of recessive pathogenic variants in individuals, determining their reproductive genetic risk. So preconception carrier screening has been very effective at reducing the incidence of severe pathologies with a strong genetic background that were endemic of specific geographical regions or ethnicities. For our patients in the IVF clinic, carrier screening reestablishes their reproductive autonomy and allows for an informed decision-making process by making them aware of the actual risks and possible alternative reproductive options, if they are affected or at risk at all. With the development of genetic technologies, this type of investigation moved from individual gene targets, specific for the population, to panels of recessive genes related to severe early onset conditions, further reducing the residual risk of the test. And nowadays, with the advent of genome sequencing, the amount of information retrieved from a single test has massively increased. And further reduction of costs in genome sequencing will make this strategy affordable and cost-effective, hopefully for every patient. Preconception genome sequencing would enable a thorough investigation of inheritable traits, including not only recessive disorders, but also polygenic diseases, as well as genetic features related to reproduction. Indeed, several causative genes have already been identified and in isolated non-syndromic cases of infertility linked to premature ovarian insufficiency, increased risk of aneuploidy, gamete maturation arrest, embryonic developmental arrest, or miscarriage. And preconception genomics can therefore even provide prognostic values for embryos' reproductive competence based on their parental genome, not only on the embryo. So because it has informative value both at the personal and reproductive level, it is foreseeable that preconception genome sequencing will be one of the technologies that will expand the most in the next few years in the context of IVF. And to conclude, it is more than likely that genomics and other omics investigative tools will become employable at different stages of conception, and that these will allow further improvements of assisted reproductive technologies by identifying inheritable reproductive risks, diagnosing extreme reproductive phenotypes at both preconception and preimplantation, provide non-invasive prognostic support to improve embryo selection, and possibly provide molecular evidence for modeling personalized IVF treatments through phenotype rescue procedures, genetic therapies, or even tailored embryo culture conditions. It can't be overstated enough, though, that important ethical concerns regarding genome sequencing and germline genetic modification will need to be thoroughly debated and clarified before any clinical application can be considered. So with this, I hope that this presentation has been helpful for at least some of you, and I thank you for your attention.
Thank you. Thank you so much, Maurizio, for your very nice presentation. Thank you, and staying in time. So we now start for the question and answer session. I will introduce you some questions that have been selected for Marcos Meseguer, and after my co-panelist, the co-chair Antonio Capalbo, will introduce questions for Maurizio Poli, and then we can discuss together. Marcos, one of the huge amount of questions you received that were selected is about the fact that various annotation relationships like extent are fascinating. However, they say, Dr. Jack Cohen, we do not see a breakdown according to maternal age. So do you conclude that maternal age is not having an effect on embryo development, or do you not comment on maternal age because the data is delivered from egg donors in your data?
Well, we have a lot of data coming from egg donation because you know that in Spain is a big part of a big proportion of our cycles is representing around 40%. And sometimes it's a very good model in particular to analyze the relevance of the selection. You know, in our side donation, normally we have a lot of embryos available, very nice, good looking blastocysts. And it's a good technology when the morphology like itself is difficult to help you to do the selection. So it's an ideal model to develop this type of model, to develop this type of selection. But of course, maternal age is affecting the embryo development. And in fact, in the algorithm of extent or in other algorithm of artificial intelligence, this information is combined in order to improve the prediction. Or, for example, in artificial intelligence, we have been testing a software that increase the prediction of miscarriage using the maternal age. So of course that is a relevant variable that can be used. But in particular in the EVA extent, we just add this information because definitely it's helpful for doing this classification. So maternal age definitely is very relevant to be used for performing any type of algorithm.
Okay, thank you. Another important topic was raised by Colin Olds and was just to clarify the higher incidence of implantation in time-lapse incubators versus benchtop. Is it attributed to the incubation environment or the algorithm which you employ on morphokinetic, or both?
Well, we may question this in the past when we were using the box incubators, and it was part of the problem of all the randomized trial that we did, because at that particular time we were still using box incubators around 2012, whatever. But then we did a transition with the benchtop incubators, and I personally believe that the quality of those incubators by itself is very good. They are small incubators, control temperature surfaces, small chambers, quick recovery. So I don't think that it's a question of the incubation by itself with the benchtop incubators. I strongly believe that it's a question of selection in our environment in Valencia, in which we have last generation benchtop incubators. So I don't believe that is, I mean, actually, that is a question of the incubation by itself. Is because the incubators are very similar in technical characteristics.
And linked to this answer, there is another question that is completely connected. And is which kind of time-lapse incubation system is preferred by embryologists in your unit, and if it's different, why?
Well, we have everything. I mean, there are people that likes one or like another, so there is not like any type of tendency. It depends of the embryologist. In fact, are relatively similar, and the point is both that we're using, that Algeria and the embryoscope have a full automation, because at the beginning, when we were using time-lapse, we have a lot of, I remember I have a lot of complaints from my colleagues because it was time consuming, and they were blaming me because we were putting the embryos in the time-lapse and it was taking longer than the other patients. But actually we have solved this problem that for me has been a relief, because actually embryologists are happy to put the embryos there because it's not increasing the amount of work. On the contrary, it's reducing, which is, I mean, we are not losing embryologists because of that, but definitely our logistics has improved since the introduction of this automation that is in all the softwares of the two time-lapse system that we have in our clinic.
Okay, and another question regarding the time-lapse system that you mentioned before is if the automated annotation by Gary is a feedback system using deep learning, and if will it improve over time or not? You hear me?
It's not using deep learning. The strategy was trained the system. It was done as software performed in the United States, which was based in the software of the EVA. And basically this software was trained to learn how to detect the cleavages, but it's not learning with the time. I mean, it's just a fixed algorithm that is used and it works. I mean, it does not need to be improved actually.
Okay. And looking more deeply into some features of the embryos that you annotated and mentioned before in your presentation, one question was about what is your opinion of blastocysts that expand and contract in fresh cycles, and if there is a relevance to implantation potential, and if you have any clue of which might be the reason for this event.
We have extensively evaluated the contraction of the embryos, and we have been spending a lot of time, the last 3 years, doing this evaluation. It was very, very interesting because when I started like embryologist, I remember my colleagues told me, Marcos, when you see an embryo that is collapsing, that's a very good sign because the embryo is very healthy. And then when we start to use this technology, we realized that it was just the opposite. So it's a bad sign, which we have no idea exactly why it's happening. I mean, we're just wondering which is a problem of the blastocyst to finally do the hatch. We know that the process is like a very massive, huge amount of energy that is used for the blastocyst to do that. So definitely we believe that it's not a good sign of the embryo development because it needs to do it. But actually it's very, very relevant. And what we have seen is that we may overcome the problem of the collapse doing assisted hatching. So we may have found an indication of assisted hatching to those that does not like to do assisted hatching to all our embryos. Forget about PGT if we are not doing PGT. That is still 80% of our cycles here in Valencia. We may find a good indication to do assisted hatching in those blastocysts that are doing collapse because we have done a study that we are presenting this year in SRM that we overcome this decrease in implantation potential by doing assisted hatching.
Okay. Did you find any correlation with also the number of contractions that the blastocysts can encounter during the expansion or not?
Yes, yes. I mean, 80% of the blastocysts that collapse, around 25% of the blastocysts collapse. From those 25, around 90% only collapse once. So it's the most typical thing. So it's less frequent to have more than one collapse, but the more collapse, the worse outcome.
Okay. And do you think, this is my curiosity, do you think that it could be a mechanism of weakness of the trophectoderm junctions that are yet present in the blastocyst, or can be generated by the procedures of towing? Can you see in the fresh, more in the fresh than in the towing blastocyst?
Well, it's interesting because in the thawed blastocyst, we see also collapse, but the collapse in the thawed blastocyst, we have been unable to link with the outcome. I mean, we have one of our PhD students that did an extensive evaluation of collapse of the warm blastocyst, and we weren't able to find this link. So if this is relevant or is important, is only in the fresh cycle, not in the warm one. So that information may be useful to understand why this process is relevant and where is the reason behind.
Very good. I guess we have time only for other questions for Maurizio. I'm sorry because you had many other questions to answer, but from the panelists you will have the list of questions and then you can answer after. Now Antonio Capalbo will ask some questions to Maurizio. Thank you, Marcos.
Thank you, Marcos. Thank you, Ledlo, and thank you, Maurizio, for very great presentation, I think, on my side. So we had also a lot of questions coming through for you from the participants. I will start with one question, which is purely embryological. So the participants would like to know, with the development of these omics strategies that may require like single embryo culture, they want to know what about what we can lose with about the group culture that in some cases, there is some level of evidence showing that they may be beneficial for embryo development and quality. So if you can comment on this.
Sure. Yes, if we want to assess non-invasively culture media, for example, we need to culture the embryos individually. There is no way to do it in group culture. However, if we can optimize our strategies, we could identify what are the factors that good embryos produce and that we may actually be able to supplement them to the ones that are lagging behind. So surely, yes, we're going back to single embryo culture if we ever move to group culture. I know some labs have, but if we want to use non-invasive, whatever type of non-invasive technology, yes, it's individual culture.
Yeah, and do you think that there will be any potential, I mean, loss of
loss of
embryo,
competence,
yes,
or
in embryo development
support,
or quality? Yeah, do you have any? Can you comment further on that?
Well, there may be some paracrine effect, culturing the embryos altogether. It's very difficult to assess, but I think there has been some studies showing it. Nonetheless, if we can characterize the proteomes of the embryos that actually go on and develop into blastocyst or even implant after transfer, we can identify and compare these profiles with the ones that arrest or do not implant. We can pinpoint what are the factors, I'm talking about protein factors or metabolites, that may be necessary for the embryos that are lagging behind. And that's what I was talking about, tailored culture environment. So supplementation of proteins or different tweaking of the incubation systems just to tailor the patient itself.
Yeah, understanding a little bit more in the basic biology.
Basic biology, of course, we need a huge amount. We're not talking about big data, we're talking about huge data here.
Yeah, and another recurrent question that I will try to summarize is, since you are probably the pioneer of the blastosynthesis approach, and they all want to know your tips and tricks on where and how, what is the best time and moment to collect this sample. We know that, for instance, a main difference with genetic analysis that is fixed on genetic variation, apart from the elephant that it deserves much wider debate. But in general, genetic abnormalities and defects are fixed through development, so are inherited and they are, fixed through development. While all the other parameters, physiological parameters that we can assess are constantly changing through development. So this can be probably one of the reasons why we have so inconsistent data from different studies with analyzing the blastocyst fluid.
Yeah,
this is what some question
we're talking about DNA, right? DNA in the
DNA, or potentially we can extend this to other biomarkers.
Omics.
Yes, to other omics. Yeah.
Yes. Well, for what it concerns DNA, I believe that the DNA in the blastocyst is highly degraded, and I think the more fragments you have in the blastocyst, the more signal you get for your DNA amplification. So that is one parameter that could affect the results that different labs may have from blastosynthesis for DNA. In terms of proteomics, I don't know many labs that perform this, and surely the data are not cross-evaluable. It's not easy. There are really little data available to cross-compare different labs. And if we could help, I believe that if there's a decision to make whether to do DNA assessment on the blastocyst or on the media or on both, I believe that the more sample, the more specimen you have, the better amplification you could have. But how the results could relate to the actual karyotype of the embryo, I doubt there's a good answer there.
And last question, Maurizio. Based on your knowledge and on this field, can you give us some kind of timeline? When do you think that, like, metabolomics or proteomics analysis from culture media or blastocyst can start to become a reality in our field for embryo selection?
Yeah, sure. I believe that a lot of work from the technical point of view, so development of mass spectrometers or NMR, has been already done, and it will continue to be done, because proteomics and metabolomics are finding applications in clinical medicine, irrespective of reproductive medicine. So that will keep going, that will keep happening. How long it will take to make it possible in IVF? Unfortunately, it's an answer that has to go back to finance. For startups, it's usual to say the more you invest, the better is going to be your product. So in the recent years, and the more it's going to work. So in the recent years, there's been huge investment in genomics, and we can see the results because there's a lot of application going on for genomics. On the other hand, and genomics may attract around 95% of all the resources, financial resources available, okay? The other 5% needs to be split to all the other omics. So I believe that there is a will somewhere, but it needs to be, resources needs to be allocated to these different omics. Difficult to say how long it will take for convince the people involved to back this up. So I would, to make it shorter, I believe 5 years we could have some kind of a clinical application because of the extensive technological validation and also all the studies that need to be required before you can actually propose a clinical trial on this.
Okay. By the way, the good news is that these clinical trials and randomized studies in this like non-invasive environment will be much easier to be carried out compared to, for instance, what we had to face with
yes, let's say that consenting patients, yeah, and ethicals are much simpler compared to invasive procedures for sure.
Okay, thank you.
Okay, yes, there are also other questions. However, we would conclude this session with a question for both of you, question from Jack Cohen, and he wrote, does it not seem that in the end, many of these new testing possibilities mentioned by both of you will result in embryo endpoint of assessment that can be used as targets for improving artificial intelligence? Is it not likely that artificial intelligence will be the dominant system for 10, 20 years from now because of its ability to cross-reference? Is for both of you.
Well, with the permission of Maurizio, I think that artificial intelligence actually is already used. We don't need to have 10 years. Actually, we have, you know, all the systems that we have available commercial. The softwares are using artificial intelligence, at least still maybe a primitive version, that is doing automation of the embryo evaluation. So it's already used. So I think it's clearly a reality. And on the contrary, and independently of the other type of technology, this is relatively easy to use. It's just a question of software. We don't need hardware. So it's cheaper and it's access to everywhere worldwide. I mean, you can access online to a website, upload your images, and then get the feedback. So it's something very easy, and I think this is already here. There are so many companies working, of course, that we need already is to have some kind of clinical studies to confirm this data. But even that we are not going to do randomized trials, there are it's quite evident that is able to replace or to at least do the same things that we do in the lab, in a similar way with similar quality.
Marcus, may I ask you a question?
Yes.
Don't you think that different baselines from different patients may alter certain morphokinetics phenotypes that you see, and not taking into considerations these baselines, whether they're genetic or proteomic? There is, it's decreasing the accuracy of the algorithm, but even across labs, I mean, all labs work at 5% or 6% or 20%, but isn't there something that is specific of the lab that is not taken into consideration by artificial intelligence? And there is no way to evaluate this through a picture?
For sure, but even, I mean, I haven't time to go through this, but we have been combining by artificial intelligence the information of the media for example, the proteins in the media. We have performed studies of proteomics. It would be great to use the non-invasive information, also combine it with artificial intelligence, because definitely all this combination increase the accuracy. But independently of that, the combination of many information will increase the accuracy. One thing that is very obvious is that the artificial intelligence is able to classify the embryos in the same way that we do. I mean, A, B, C, D, E, following ASEBI, for example, or SIERR classification. And that's a clear advantage because we know that between the embryologists, we have a lot of heterogeneity in evaluation. So it gives consistency to only to the morphology by itself. Forget about
But what you define here as artificial intelligence, you mean automated scoring of the embryo. Okay.
Okay, sorry to interrupt this discussion, but I know it's really interesting and we can go on. But I have a really important question to ask, and this will be the last one for tonight for Marcos. They are asking, why did Spanish food never become as popular internationally as Italian food?
This is not true. You can come to Valencia, the best players in the world are here. The best rice. Risotto is
nothing. No.
Marcos, you are in the wrong place to say that.
Valencia invented
we invented the paella.
The paella, no?
I talked to some Spanish friends and they say Italians do better marketing.
Okay.
We are very bad marketing, very bad. Very bad.
Okay, thank you everyone. And I think we can now conclude our session. It was a pleasure to organize this meeting with a great team from SIERR. I want to thank them along with two excellent speakers, Marcos and Maurizio. Thank you. And thanks to the two moderators, Catello and Antonio, who contributed to a very interesting discussion. I would like also to thank the host from IVF meetings, Giles, Jack, Marianne, Thomas, Peter, and Alison. And before ending this session, I would like to mention you that there will be an upcoming session next week on Thursday, the 16th of June. And the webinar topic will be PGTA and the IVF lab, improving outcomes and techniques. This will be held by Dr. Harton, Dr. Nagy, Dr. Kirk, and Dr. McCullough. So thank you all for participating, and for those that are close to Italy, good night. For the others, good afternoon, and enjoy your rest of the day. So thank you, everyone.
And ciao.
And ciao. Buonanotte.
Buonanotte.